Hello, science fans! Welcome back to our fascinating exploration of the microscopic world inside your body. Today, we are diving into one of the most amazing and essential processes happening inside you right now. Something that keeps you alive, growing, and thriving every single moment. Imagine your body as a bustling city made up of trillions of cells, each one acting like a busy factory, working around the clock to keep everything running smoothly. Last time, we explored the process of transcription, how your DNA, the master blueprint, is carefully copied into messenger RNA, or mRNA, inside the cell's nucleus. mRNA acts like a courier, carrying those vital instructions from the protected nucleus out into the bustling cytoplasm, where the real action happens. But just like a recipe book isn't a cake, a set of instructions isn't useful unless something can read and follow them to create something new. That's where translation comes in. This is the step where the cell reads the mRNA and actually builds a protein, turning information into action. Translation is a lot like a chef using a recipe to bake a cake, carefully following each step to get the perfect result. This incredible process is happening in trillions of your cells every second, building everything from your muscles and skin to the enzymes that power your metabolism. Translation means converting the language of mRNA, those 4 letters A, U, G, and C, into the language of proteins, which are made up of 20 different amino acids. Your cells read the mRNA code and with incredible precision, assemble amino acids in just the right order to make each unique protein your body needs. Without translation, all the information stored in your DNA would remain locked away, never used, and life as we know it simply couldn't exist. Translation is the vital bridge that connects your genetic code to the living, breathing functions of your body. So let's meet the incredible team of molecular machines that makes this amazing process possible. To truly understand translation, let's meet the remarkable cast of molecular players that bring this process to life inside every cell. First, there's mRNA, the messenger RNA. Think of it as the instruction manual, carefully written in 3-letter words called codons. Each codon is a unique combination of 3 nucleotides, and each one codes for a specific amino acid, providing the blueprint for building a protein. Next up is the ribosome, the cell's molecular machine. It's like a highly specialized factory that reads the mRNA instructions and assembles the protein, step by step with incredible precision. Imagine the ribosome as a workbench, moving along the mRNA, reading each codon, and connecting amino acids in the exact order dictated by the genetic code. Then, we have transfer RNA, or tRNA, the delivery trucks of the cell. These molecules are responsible for bringing the right amino acids to the ribosome at just the right moment. Each tRNA has a special region called an anticodon that matches up with a codon on the mRNA, ensuring that the correct amino acid is delivered for each step of the process. For example, if the mRNA codon is UAC, the tRNA with the complementary anticodon AUG will bring the amino acid tyrosine to the growing protein chain. And finally, the amino acids themselves, the essential building blocks of proteins. These small molecules are the raw materials that get linked together to form the final product. There are 20 different types of amino acids, each with its own unique shape and properties, like a set of colorful Lego bricks that can be combined in endless ways to create countless proteins. The ribosome reads the mRNA, tRNA delivers the correct amino acid, and the ribosome links them together, forming a growing chain that will become a functional protein. The process begins at a special start codon, AUG, which signals where to start building the protein. When the ribosome encounters a stop codon, it knows the protein is complete. It's a perfectly coordinated team effort, with each molecule playing its part in harmony to ensure accuracy and efficiency. Each player has a unique and essential role, but together, they transform genetic information into the proteins that power life itself. That's the cast of translation. Now let's see how the show begins. On to initiation. Every translation story starts with initiation. The ribosome's small subunit attaches to the mRNA's beginning, scanning for the start codon, AUG. AUG signals where to start and codes for methionine, the first amino acid. The initiator tRNA, carrying methionine, binds to AUG. This sets the reading frame and signals the machinery to get to work. The large ribosomal subunit joins, forming the complete ribosome. Now, the mRNA, ribosome, and first tRNA are all in place. The stage is set for protein construction. Initiation is complete. The engine is primed. Next up, elongation. Now, elongation begins. The protein chain is built, one amino acid at a time. The ribosome moves along mRNA, reading codons and adding amino acids. tRNAs bring the correct amino acids to the ribosome's A site. The ribosome links the new amino acid to the growing chain at the P site. A peptide bond forms, connecting the amino acids. The ribosome shifts forward, moving tRNAs and exposing the next codon. The empty tRNA exits, and the cycle repeats. This process continues, rapidly building the protein. Each step is precise, ensuring the correct sequence. Elongation is the heart of protein synthesis. Eventually, the ribosome reaches a stop codon, UAA, UAG, or UGA. These codons don't code for any amino acid. No tRNA matches them. Instead, a release factor protein binds to the stop codon. This triggers the ribosome to add a water molecule, releasing the finished polypeptide chain. The new protein floats away, ready for its next steps. The ribosome and mRNA separate and can be reused. Termination ensures the protein is released at the right moment. The translation machine resets for another round. The cell efficiently turns genetic code into functional proteins. The journey from mRNA to protein is complete. The new polypeptide isn't a working protein yet. It must fold into a precise 3D shape. Folding begins immediately, guided by the amino acid sequence. Some parts attract, others repel, creating twists and bends. The final shape determines the protein's function. Shape is everything. Helper proteins called chaperones assist in proper folding. Some proteins work alone, others join with more chains, like hemoglobin's 4-part structure. After folding, proteins may get chemical tags that guide them to their destination. From a simple code, we get complex, functional machines. This is the central story of molecular biology. The dance of synthesis and folding makes life possible. Why go through all this effort just to make proteins? It might seem like a lot of work, but there's a good reason for it. Proteins are the true workhorses of your body. They do almost everything that keeps you alive and healthy. They provide structure and support. Think of actin and myosin in your muscles that help you move, keratin that gives strength to your hair and nails, and collagen that keeps your skin firm and flexible. Enzymes, which are special proteins, speed up the chemical reactions that keep you alive, like breaking down your food so you can get energy. Other proteins act as messengers, such as insulin, which helps control your blood sugar, and transporters, like hemoglobin, which carries oxygen through your bloodstream to every cell. Channel proteins in your cell membranes act like security guards, deciding what can enter or leave your cells, keeping everything in balance. Every single function, from your muscles moving to your brain sending messages, to your immune system fighting off germs, depends on proteins working together in harmony. And it all begins with that mRNA message and the amazing translation process that turns genetic code into life. Next time, we'll explore what happens when the message isn't quite right, when there's a typo in the code, known as a mutation, and how that can change everything. Science is truly amazing. Thanks for joining this journey from gene to protein. Stay curious and see you next time.